record this for you, right? So you can actually you can come back to this if you need. When you're writing your coursework, you can then review this. You can look at the guidance from this document, okay? Okay, so Lewis, Leo, if you just want to, yeah.
Should we write down the services? Or?
No, that's fine for now. If you can just take your seats and get the guidance documents out, the Group 4 IA, Internal Assessment Group 4 guidance out. So yesterday we went straight to the back and we were talking about the TOK stuff. So I just want to finish that if possible. Just to be aware that this is here. So there's a bit of a blurb about the scientific method. So I'm on the last page, right at the back, on the Group 4 and TOK. So a bit of a blurb about the scientific method, and I was highlighting to you guys that your coursework. I don't like the fact that I've got two backs in my face. Could you guys come and sit around here? Don't worry, you won't be on the screen. I want to be able to see your faces. Lewis, could you mind coming over here so that I can actually I want to feel like I'm having a conversation with you as opposed to, you know. Thank you very much. Okay, so this blurb about the scientific method, this is obviously I've included this for your benefit of your TOK presentations and essays, but also just so you're aware that what you're doing in your coursework in science, biology, is that you are effectively mimicking this scientific method, okay? And that's the context of why the IB recommends that you do this individual investigation, okay? For your work, you can do you don't actually have to do an experiment, okay? If you wanted to look at a database and do some sort of statistical analysis of a database, you could do that as well. Obviously, experimental work is the kind of what most people tend to do. Yeah.
What would you say? I don't want to say easier. What would you say is better to, I guess, to do a write-up about, or just to do like an experiment or analyzing database? What would you suggest?
Depends what your strengths are. If you're a good mathematician and you understand statistics, then a database could be a good option. I mean, you'd still need to have a research question and a hypothesis background, that sort of stuff, and you still need to have a method of, you know, what database and how you need to explain how you actually queried and analyzed the data. But if you like crunching numbers, if you're like me, then that would be a good way to go. And there are lots of biology databases to do with genomes that you can actually look at, and I've got some guidance on that as well. So don't be limited. You could look at a database. You could also design your experiment around a simulation as well. There are some, like photosynthesis, we'll look at those when we get onto photosynthesis. There are some simulations, software that you could use to run your experiments. You don't necessarily have to do a wet practical, but yeah, you still need to kind of follow this kind of procedure of the scientific method, okay? So I just wanted to, on the last page of this guidance then, just really quickly before we kind of get into the design part, just to be aware of these terms. We will look at natural sciences in terms of TOK, but there's kind of key themes in science of this idea of modelling, okay, so that scientists actually build with simulations, with databases, in physical reality, we actually build models. And what do we mean by a model in science? We tend to mean something that is a simplification of reality, okay? So when we study the structure of DNA, you know, we present it as a model. When we talked about the phospholipid bilayer, right, of cell membranes, we present that as a diagram. That diagram is a model of a simplified model of what we think is going on. And in science, what we try to do is obviously test those models to give, you know, a scientific model should make a prediction, allow you to make a prediction about something, and you can test those predictions to help you find out whether that model is correct or not. Okay, to refine it. Also, this idea in science of falsifiability. So, as I said, Karl Popper is a philosopher of science, basically in the now I'm really stuck, I think 1940s, I need to check actually, basically came up with the idea that, you know, really for science to be considered science, it needs to be, the knowledge needs to be open to being shown to be wrong. Okay, so anything in science can be, knowledge in science can be changed. It does change over time because experimental data shows our theories to be incorrect. That is quite often, people tend to say, oh, science is unreliable because it changes. Well, actually, it's one of its greatest strengths. It's not dogmatic. It doesn't say this is how things are and this is how it always is. Knowledge in science tends to be flexible in the sense that we're trying to understand as best we can what reality around us is actually like. And that final term there, paradigm shift. So this is the idea that we have in science. We have an understanding about how something works, whether it's the phospholipid bilayer. We understand that's our paradigm. That's our view of how cell membranes work. But over time, with new evidence, that may change. So in science, we have these things called paradigm shifts. This idea that the view of science over time does evolve and does change. Okay, and that's quite a good thing to contrast with, maybe kind of religious knowledge systems, which tend not to do that. Okay, so just be aware that there's some advice for TOK kind of at the back of this document. Okay, let's get on, because I do want to give you some time to actually plan this experiment. So we are going to look at, in your IA, so I'm going right back to the beginning now, okay, right back to the very beginning, first page, and I said there were these criteria on personal engagement, exploration, analysis, evaluation, and communication, okay? We are going to be looking now at the exploration criterion. We've looked at the analysis, we've looked at the evaluation, we're going to look at the exploration. And for standard level students, this is the first time you've done it. Higher level, we did already kind of go through this, but I think it's worth you hearing this again. So I'm going to move on to look at the page, I think it's page number 3. What is the exploration criterion assessing? So this is assessing the extent to which the student, that's you, establishes the scientific context for the work, states a clear and focused research question, that's what we talked about this morning, and uses concepts and techniques appropriate to the diploma program level. Where appropriate, this criterion also assesses your awareness of safety, environmental and ethical considerations. So with that practical we've just done, were there any safety issues that you needed to be aware of?
Hot water.
Yeah, okay, so hot equipment, there's a burn, or even with water, with steam, there is a scalding risk, okay? What else?
Chemicals we were dealing.
Yeah, okay, so hazards of iodine, how would you be able to find out what the hazards of those chemicals were? It's usually written on the bottle, so you can look at the bottle. There are also, we have, and you'll need to do this for your actual write-up, there are, if you're going to use any chemicals, there are what we call HAS cards that Miss Hartley has, which you can check to find out what the actual safety precautions for a particular chemical are. You will need to check that, and you will need to put a risk assessment together in your exploration. Okay?
Can we get some examples, maybe? Because I'm not really 100% sure.
I'll show you. There is one in this document. Okay? You also need to be aware of any ethical issues. If you're doing any experiments that involve humans or animals, you need to be aware of what potential ethical issues there might be. Can we think of, if you're going to test, if you wanted to do an experiment that involved human subjects, what would be the ethical issues with that?
Keeping our names private.
Okay, so confidentiality, yep, definitely. Pardon? Not inflicting physical pain, yep. What else?
Telling them what you're going to do beforehand so they have informed consent.
Good, they have to have informed consent. They need to know and understand what it is you're doing, yes. They need to be able to withdraw them at any time as well if they want to, okay? You need to also let them know what you're going to do with your data. Yeah, that's also very important too. With animals, we need to think about, are we causing any unnatural stress for that animal or, organism? We doing something that would be outside of its normal environment, okay? And you need to then actually think that through if you decide to use any live organisms for your practical work, okay? And there are actually some IB guidelines for animal experimentation, okay, if you decide to use that. Okay, what about environmental considerations?
Something that can damage the environment.
Yeah, good. So what kind of things could damage the environment? Yeah, so thinking about the chemicals you're using again, are there any environmental concerns in terms of disposal? We had a student this year in grade 12 use animal tissue, hearts from different animals, okay? How do you dispose of that material? If you're working with bacteria, how do you dispose safely of that material without there being any health or environmental impact? Yep.
Don't you have like the bio waste?
Yeah, good. So again, I will with each of you talk individually if that's something you want to do, what procedures. But normally, yes, with bio waste, we'd have a coloured bag that said biohazard on it. All the materials goes in there. It would either be burnt or there's something called an autoclave, which is basically like a pan that you can put things in that heats it really hot and steams it as well. So it basically takes up to 120 degrees and steams whatever's in there. So it basically kills anything that would be growing on there. Okay. So yeah. So just be aware that that is one of the things you're being assessed on. Okay. So we've got. So this criterion is worth 6 marks. Okay. And there are 4 strands. Okay, that you're assessed on. So for each mark descriptor, 1, 2, 3, 4, 5, 6, there are 4 strands. So if we look at just strand 1 is about the research question, okay, and the topic of the investigation. So a level 1 to 2 piece of work will identify the topic and a research question, but it's not very clear or focused. Okay. To get 3 or 4 marks for that band, it's got to be a bit clearer. It's got to be a relevant research question, but again, your research question wouldn't be fully focused. So this is what I was talking about, this idea of making sure that it's very clear what your dependent and independent variables are. What are you actually measuring for your dependent variable? What are you actually changing? What are the units? That needs to be very clear in your research question if you want to get up to the 5-6 band there, which says the topic of the investigation is identified and a relevant and fully focused research question is clearly described. Okay. The second strand is assessing the background information you provide. Now, what do we mean by background information? Want to know why this investigation is relevant? Why bother doing it? But also, what's the science behind it? What have you learnt in your diploma course? Okay, and what's the context? What's the scientific context? So when you're designing experimental enzymes, I'm expecting you to be writing there about how enzymes work, enzyme substrate specificity, how enzymes are affected by whatever environmental or whatever physical variable you're going to be manipulating, whether you decide to design an experiment on substrate concentration or on temperature. Actually, in fact, I'm going to rule temperature out because we've just done an experiment on it. So substrate concentration or pH. Okay? How do those things affect enzyme activity?
Did we do pH last year?
I don't think so. No.
We did something last year.
We'd probably do temperature. I normally just do temperature.
Yeah, a little bit on pH, but that was it.
Yeah. We talked about it on yesterday, the effect of pH on enzymes. Yeah.
So we can do an investigation on pH
Yeah.
level.
You're not going to carry this one out. You're just going to design it, okay? And I'll give you some feedback on it. You're not actually going to carry this experiment out, so don't worry about that per se.
So it's not a full lab report?
No, it's just the exploration. We're just doing the exploration. So you're getting a chance to do the exploration, design an experiment. I'll give you some feedback on it. Now, you may decide this is something you want to do for your IA, and that's fine too. So this could be, something you use for your IA.
Oh, we could use this, not the same. We could use the same explanation.
Yeah, there's no reason why you couldn't. If it's independent enough, right? And I need to talk you through that. Okay, we'll discuss that individually. Okay. Okay, so background information. Then the third strand is looking at your methodology. So what are you looking at? The 5-6 descriptor for strand 3. The methodology of the investigation is highly appropriate to address the research question because it takes into consideration all, or nearly all, of the significant factors that may influence the relevance, reliability, and sufficiency of the collected data. What that's basically saying is, have you controlled all of the other variables that could control, and does your method make that clear? Okay, So if you're going to change pH, you need to think about the substrate concentrations, the enzyme concentrations, the temperature, okay, and any other variables you think of that might influence enzyme activity. And then finally, the final strand, strand 4, is looking at your awareness of the safety aspects. Okay? All right? So on the next page, then, there's some specific guidance for personal engagement exploration. So basically, these are kind of subheadings you should use, I would suggest. So you want a title. The title is not the same thing as the research question. Okay? The title for this might be an investigation into the effect of pH on enzyme activity. It's not the same thing as your research question. It needs to be descriptive. Okay? Nice descriptive title. Makes it... You get marks for the presentation of your work as well. So be professional, right? This is your report. Like, what are you've got to submit it to the IB. You want to take some pride in that. Nice title. Your research question we've discussed. I'm not going to say anything more about that. It needs to be phrased as a clear question, and it needs to be, make it clear what your independent variable are. So within your background information, again, this is a bit more difficult for you, but you can explain why you've chosen either pH or substrate concentration. But when you actually carry out your investigation, this will be the chance for you to really talk about why you've decided to go with a particular topic. Why is it personally significant to you? Okay, so this section, the background information, should explain to a reader who has some background in biology the information needed in order to understand the significance of your investigation and appreciate the importance of your results. Explain why you've chosen your independent variable, and focus on your chosen independent variables and research question. And obviously reference anything. I would suggest you are referencing your course textbook. If you're referencing Wikipedia or some random website and not your course textbook, I'm like, why? Why would you not use your course textbook as your first point of call for a reference? Okay, because that's tailored for your course. And it's an IB examiner who's going to be marking your work. Yeah.
Wait, so if most of our, resources are our book, it's fine.
That's fine. Yeah, it doesn't. You're not getting marked on how many resources you've used, but you do need to reference your theory, and your textbook would be the best place for that. Okay, hypothesis isn't formally required. We talked about this yesterday, but you should really include it. Your hypothesis then is just a general explanation of the science and what you think generally should happen how does pH affect enzyme activity, and then you're going to make a prediction specific to your investigation. Okay, so as I change, the pH of pepsin or amylase, I would expect to find this result. So specific prediction.
Without explanation, just prediction.
Just a prediction, yeah. The hypothesis is kind of the explanation. Then you go on to variables. You've got to make it clear. You can put this in a table if it's easier. So independent variable, dependent variable, and then your controlled variables. Okay, how are you going to control your controlled variables? If you can't control it, like room temperature, how do you control room temperature? You can't really. So you're going to monitor it. You might have a temperature probe that's going to keep an eye on what the actual temperature of the room is while you're conducting your experiment. You can at least monitor the temperature. Okay, if you can't control it. Then your materials and apparatus list, and then safety. So this is on your next page, Lewis, page 5. There is a breakdown. There's an example there of what a risk assessment would look like. So a table again saying what the hazard is, what the risk is, how you can minimize the risk, what potential emergency action you would have to take, and is it a high, medium, or low risk. So burning in that experiment, probably a medium risk if you're working at 70 degrees. Cutting yourself with breaking glass, probably low risk. There's a high chance of it happening, but it's not going to necessarily kill you, right? Do you know what I mean? So yeah, low, medium, and high level of risk. Okay.
Bunsen burners, would that be?
Bunsen burners, yeah, high risk of burning. Yeah. Preliminary experiments, you don't have any for this design, so you can skip this for now. But for your actual investigation, you will conduct preliminary experiments. So you're going to basically what that means is you'll design a method, you'll test it, and then you'll change it depending on you know, because it's very well writing a method and then finding out actually it doesn't work very well, right? So that's your preliminary experiment, and you'll have an hour or 2 hours in which to do your preliminary experiments. Okay.
Does this mean you have to make, like, for example, in your method you have a list of different things that you're going to do? So you just write the same list but with the changes in?
Yeah, you would adapt it, and then in your write-up you're going to explain how you adapted it.
Okay.
Okay, you just say, well, I carried out this experiment, found out this didn't work, so I decided to do this instead.
Okay.
And that demonstrates personal engagement to your examiner. That shows that this is a student who's actually actively thinking about, like a scientist, how do I solve this problem? I'm going to try and answer a question. How do I go about doing it? Okay, it's more than likely that most of you won't be able to answer your questions, right? The point is, how do you try and problem solve like a scientist would? Okay, in 10 hours. You've got 10 hours of class time for it. Okay, so there's a time constraint. That's fine, but you've got to demonstrate the ability to problem solve. Okay? Yeah, and then finally, your method is the last thing, and there's a list of steps here to tell you what to do. All right? So is that fairly clear?
This is all for the exploration.
Just for the exploration, yeah.
So
The full lab report is going to be...
Full lab report is limited to 12 pages.
Yeah, that's going to be the biggest problem. I
Yeah.
think, isn't it?
Yeah.
so 12 pages front and back, or 12 pages?
12 pages.
So it's not even a word count, it's just 12 pages.
12 pages, yeah.
So it doesn't matter how much you write, just how.
If I make it like a
Oh,
It's got to be 12.
12 font, yeah.
Yeah.
12, minimum of 12, size 12. Yeah.
You could,
Arial, yeah.
Um,
12 pages. Conciseness. Conciseness is one of the things you're getting marked on for communication. Your data, you don't need to present all your raw data. You can just present summary tables of your means, standard
You
deviations.
don't have to have all of them.
okay.
Okay. Yeah.
Also, for the end of the method, include a control group if applicable.
Yes.
So, like how would
So
you-
control group would not be applicable necessarily. It depends on what topic you're studying. If you're looking at biochemistry, control groups probably are. If you're doing something in ecology, okay, which we will have covered by the time you do your experiments, you know, in ecological investigation, it's very hard to have a control. You know, you use statistics to try and instead. But yeah, so I will advise you on that. Yeah, we'll talk through your investigations individually. Okay. But yes, for your pH, for your enzyme experiment, you should have a control. Yeah, you think what that control is. Okay. Any questions, boys? No? All right, I'm going to stop that.